Hello, my name is Gerard Hurwitz, and I was professor at New York School of Medicine. And in 1960, I discovered RNA polymerase. But before I discovered this, Avery found that DNA is what contains heritable information. And we wanted to find out, how does this DNA get transcribed and then translated into physical proteins? And it turns out that there's an intermediate step where RNA is involved, and there's a lot of enzymes involved in this process along the way. Let's start out by just looking at DNA. DNA is a structure of sugars and bases in a double strand-like shape. There are 4 bases: adenine, thymine, guanine, and cytosine, and it's structured in a way that is called complementary base pairing, where when you have a guanine, it's directly across from cytosine, and when you have a thymine, it's directly across from an adenine, and they are hydrogen bonded together. The sequence of these bases is that heritable information that I was talking about. And specific sequences of these bases are called promoting sequences, and they are recognized by proteins that will tell RNA polymerase where to start transcribing the DNA into RNA. So the RNA polymerase comes in and it attaches to the antisense strand, and it transcribes from the 3' to the 5' end of this antisense strand, producing RNA starting at the 5' to the 3'. And then this RNA gets taken and made into proteins.
Whoa, Gerard, you're forgetting about the methods of transcriptional control. For those of you that don't know, I'm Francois Jacob, and I found Black operon model in 1961 with my partner, Jacques Monod. So back to transcription. You're forgetting about the genetic switches that can be present, and that can be either positive or negative regulation. Now, when there's an activator, which is transcription factor bound, this allows the RNA polymerase to bind and transcribe the DNA. But if the ligand is present, it can bind to the activator, disassociate itself from the DNA, and now the gene is completely turned off and cannot be transcribed. In the form of negative regulation, a repressor can be bound or does not allow the RNA polymerase to bind and transcribe the DNA. But when a certain ligand is present, it can bind to the repressor, disassociate itself from the DNA, and allow RNA polymerase to bind. And that's just one method, transcriptional methods of control.
Thanks, Gerard. Hey everybody, I'm Dr. Ricardo Roberts, and I was in Cold Spring Harbor with Jim Watson in his lab in the 70s, where I worked with Phil Sharp in order to discover 5' end capping and RNA splicing. So what we found is that after trying to sequence these RNA using Sanger sequencing, that there was a conserved end on every mRNA that came out into the cytosol, being this 5' cap. Before that, mRNA has to undergo some conformational changes using a spliceosome, which then cuts out these non-coding regions of RNA, like this, but with less work. Please. And then ligates them all back together in the right order, like I just did. Then it adds on this 5' end cap and this poly A tail on the 3' end, which helps in degradation during the cytosolic part of its lifetime. And then it gets sent off into
Hold on,
the...
Ricardo. You're forgetting about post-transcriptional forms of regulation. Now, for example, this could consist of alternative splicing. So when you cut out all of the introns in the mRNA, sometimes it'll only cut out one of the 2. So it can cut out, instead of when they're all being trying to be ligated together, you'll only have alternative splicing, where it cuts out one intron. Let's see if I can do this. But it'll leave the other. And this allows for different proteins to be made from the same sequence.
Very interesting.
Yes, learn something new every day.
You do.
Hello, I'm Dr. Francis Crick, and my colleagues and I in 1961 discovered the genetic code. The way we did this was using bacteriophages, and we used a mutagen called proflavin to cause a one base pair frame shift in a gene that would then often not be expressed in the bacteria due to that one base pair change. We then tried 3 base pairs and found that it often had little to no effect in that expression, and that we would see the protein made. So this led to the first discovery that the genetic code is actually based on a 3-sequence codon. This finding sparked subsequent experiments in which other scientists found the 64 permutations of that 3 base pair code, which allowed for redundancy because there are not 64 amino acids, but no ambiguity. This process of translation for the genetic code begins when the small ribosomal subunit, as it moves down the messenger RNA, is able to identify the start codon, represented by AUG. You then have tight binding, just like that, after which time the initiator complex that helped it to locate that position bind falls away. Translation will then continue as the next tRNA with the appropriate amino acid identifies the active site, or the amino acid tRNA, binds to that complementary sequence. Also associated with it is elongation factor TU, as represented by this protein. Everybody knows this is the proper appearance of a protein, as displayed in every scientific textbook. This happens to be elongation factor TU, associated with the tRNA. And when it is hydrolyzed, because it also has a bound GTP, when it's hydrolyzed, as seen here, also shown by every textbook, yellow spheres, it falls away. The energy associated with that GTP hydrolysis and the conformational change of that elongation factor is what allows a new amide bond to form between the amino acid on that tRNA and the growing polypeptide chain, as shown here. To continue this process, another protein, elongation factor G, which is obviously totally different from the first one, and has great affinity for the A site, binds to the complex and causes the large unit to shift down by 1 codon to encompass the G protein. Finally, that G protein is hydrolyzed, and that conformational change in the protein causes the entire unit, including the small ribosomal unit, to shift down an entire codon position, and that G protein dissociates and falls away, thereby leaving an exposed A site for the next tRNA to bind and continue that process until the stop codon is reached and you have your final polypeptide. As this protein was made, it automatically starts to form its proper conformation due to hydrogen bonding at the secondary level with alpha helices and beta pleated sheets, with further structure given by long-range interactions between the R groups of those amino acids.
Whoa, slow your roll there, Crick.
What a
We didn't-
Watson thing to say.
We didn't even talk about methods of translational control. before we get into folding of proteins, we have to think about the ways mRNA can be affected, control methods of translation. Now, when you have your strand of mRNA, for an example, it could self-fold on itself, preventing the protein, the ribosomal subunit complex to not bind to it. Yeah, so it can form loops where that cannot happen. An example of where this is used is with heat shock proteins, where it needs to have elevated temperatures to straighten out the mRNA, allowing for the ribosomal subunit to bind and then translate the protein. Now you may continue with your methods of folding proteins.